Hello everyone, welcome to the first video of my YouTube channel. My name is Dr. Shreya S and I've just completed my MD Pathology from Kasturba Medical College, Manipal. This is us trying to find out the pathologic behind a very important process of the irreversible cell injury, necrosis. Feel free to drop your doubts, questions, comments in the comment section below. Questions apart from pathology like the existence of aliens, why is the earth round, can't be answered unfortunately, but yes, we can have an open discussion on it since knowledge shared is wisdom built. So let's start. So before we move on to understanding what is necrosis, let's try to understand what cell injury is as a process. I'll explain this flowchart like a story. So now here we have a gentleman called Mr. Cell who's peacefully meditating and he calls this his homeostatic or his steady state. But there are certain offending agents called as a injurious stimulus who try to disrupt this peaceful meditating state by either causing cell injury or by giving Mr. Cell a lot of stress. But our Mr. Cell is a very strong person. He tries to adapt himself to this injury by four methods. One is atrophy, which is decrease in the organ size by decrease of cell size. Hypertrophy, which is increase in the organ size by increasing the cell size. Hyperplasia, which is increase in the organ size by increasing the cell number. And metaplasia, which is transformation of one cell type to another cell type. And when the cell injury is mild, then Mr. Cell goes back to his meditative or homeostatic steady state, and this process is called as reversible injury. But if the cell injury is very severe, then our poor Mr. Cell, instead of resting in peace, will become rest in peace and he will undergo cell death, and this process is called as irreversible injury. Irreversible injury occurs through two important pathways. One is necrosis and the other is apoptosis. So here we have a very detailed definition of necrosis. It is defined as an irreversible form of cell death with denaturation of intracellular proteins and enzymatic digestion of lethally injured cell, with the earlier sign being increase in the enzyme levels. Phew, isn't this very extensive? But let's try to decode it step by step in the following slides. So these are the morphological changes in necrosis. First, what are the light microscopy changes in necrosis? So to the left, you can see a very normal tubular epithelial cell, and to the right, you can see that this tubular epithelial cell is photographed in his last moments undergoing necrosis. As you can see here, the nuclei are completely lost, the cell appears completely fragmented, it has no recognition from the photo in the left, and it appears to be very pink, which is also called as eosinophilia. Doesn't it look like a moth has completely bitten off the cell and disintegrated it? Which is why the necrosed tissue or the necrosed cell always looks like a moth-eaten appearance. And there is areas which appear completely homogeneous and completely glassy. What are the electron microscopy changes which are seen in necrosis? So this is a photograph of a rabbit kidney. To your left you can see that it is intact, and to your right you can see that this rabbit kidney tissue is photographed in its last moments. So as you can see here, the plasma membrane which is intact on the left has completely been disrupted towards the right, and the mitochondria which is intact here is completely swollen up with electron dense deposits. So as you can see, the body is inside the mitochondria and around it are electron dense deposits, and this suggests that the tissue is undergoing necrosis. We saw what happens to the cellular outline during necrosis. Now let's see what happens to the cell's most pivotal part, the nucleus, during necrosis. So during necrosis, the nucleus undergoes three major changes. One is pycnosis, the other is karyorrhexis, and the third is karyolysis. Now let's see, this is a normal nucleus, intact nucleus with an intact chromatin. In pycnosis, what happens is this chromatin undergoes complete condensation. It becomes clumped and it becomes very small. In karyorrhexis, this nucleus breaks apart, that is, it undergoes fragmentation, and that is called as karyorrhexis. The nucleus completely fades away. It is called dissolution, and that process is called as karyolysis. On a light microscopy, this is how pycnosis looks, that is complete chromatin condensation. Karyorrhexis is fragmentation of the nucleus, and karyolysis is fading away or the dissolution of the nucleus. So before I move into detail about the types of necrosis, this is a wonderful picture which gives you an overview of the same. Always remember that the cytoplasm will tell you how the cells have died. Coagulative necrosis usually occurs due to protein denaturation. Liquefactive necrosis will usually occur due to enzyme hydrolysis. Caseous necrosis is a combination of coagulative necrosis plus liquefactive necrosis. Enzymatic fat necrosis occurs in tissues such as pancreas, and that results in saponification. There is another type of necrosis called fibrinoid necrosis that usually occurs in the walls of the blood vessels. So let's move on to each type in detail. So first is coagulative necrosis, wherein the architecture of the tissue will be preserved. There will be no proteolysis, and the localized area is called as an infarct. Coagulative necrosis is a process that usually occurs in most solid organs, but always remember the brain will never undergo coagulative necrosis. So this is a picture that shows how coagulative necrosis occurs in a kidney. So to your left you can see the gross picture of the kidney, which shows a wedge shaped infarct. To your right you can see the light microscopy picture of the kidney, and here you can clearly divide this light microscopy into two parts. Towards your left you can see a completely intact kidney with normal glomeruli and normal looking tubules. But to your right you can see that the tubules have undergone coagulative necrosis. The architecture of the tubules, the cell outline is still preserved, but there is a lot of dense inflammatory infiltrate, and there are areas of coagulative necrosis. The next important type of necrosis is called as liquefactive necrosis. So in liquefactive necrosis, we must understand that proteolysis is present, and this type of necrosis is seen in solid organ as well as brain. And this occurs in case of a bacterial or a fungal infection. So as you can see here, this is a gross image of a cerebral parenchyma which contains an infarct. A subtype of liquefactive necrosis is called as gangrenous necrosis. What is gangrenous necrosis? This primarily occurs in the limb. So whenever there is a reduced blood supply to the limb, this limb will undergo coagulative necrosis. And if superadded bacterial infection is present to this coagulative necrosis, this will undergo liquefactive necrosis or gangrenous necrosis. So before we move on to the other type of necrosis, let us discuss the differences between two major type of necrosis. One is dry gangrene, another is wet gangrene. So what happens in dry gangrene? Whenever there is a aseptic ulceration, there is a slow gradual loss of blood supply, and the affected area skin appears dry, shriveled and mummified. There is a clear line of demarcation that is usually seen. The only advantage of dry gangrene, if there are any, is that since the affected area rests within the line of demarcation, limited amputation is sufficient. The causes for dry gangrene are atherosclerosis, thromboangiitis obliterans. In wet gangrene, what happens is there is a septic ulceration. So there is a sudden loss of blood supply and the affected area appears edematous, putrefied and discolored. Since there is no line of demarcation over here, it is very vague. This can extend proximally and rapidly. This usually occurs due to emboli and trauma, and since there is an extensive area which is affected, major high amputation is often needed. Now we move on to the third type of necrosis, which is called as caseous necrosis. Caseous necrosis, like we had discussed before, is a combination of coagulative as well as liquefactive necrosis. So herein you will have a cheese-like friable or a necrotic tissue primarily seen in the lung in case of a tuberculosis or a granuloma. So as you can see in this gross picture, this is tuberculosis of the lung, and there's a large area of caseous necrosis. It has yellow, white and cheesy appearing debris. So after we have understood the three types of necrosis, let's move on to the fourth type of necrosis, which is called fat necrosis. It usually occurs in tissues like the pancreatic tissue. So using this pancreatic tissue as an example, let's try to understand the process of fat necrosis. What happens in the pancreatic tissue? There are areas of fat destruction because of various causes. Now that will release pancreatic lipase or the triglyceride. Once the triglycerides are released, they will release fatty acid and calcium, and these create chalky white areas called as fat saponification. So now let's move on to the last type of necrosis, fibrinoid necrosis. What is fibrinoid necrosis? Like I had mentioned before, it is a type of necrosis that usually occurs in the walls of the blood vessels. But why does it occur in the walls of the blood vessel? That is because there is certain immune reaction which will lead to deposition of the antigen antibody complex. So there are certain immune reaction which will lead to antibodies forming complexes with antigens, and these usually deposit along with a lot of plasma protein which has leaked out of the vessels. So like we saw before, the plasma membrane usually bursts and all these plasma protein leak out. Them together with the antigen antibody complexes, they deposit themselves in the walls of the blood vessel. So there will be a bright pink and amorphous appearance in the H&E stain, and this is called as fibrinoid. Are you confused? I will move on to the next slide to show you better. So as you can see here, this photo shows the wall of an artery which has a very bright pink area of necrosis. Why is this bright pink area present? Because of antigen antibody complex deposition along with plasma protein. And what is this bright pink amorphous substance called? It is called as fibrinoid. So this was a brief discussion that we had about necrosis. But do you all remember in the initial flowchart I had discussed about one more process of irreversible cell injury called apoptosis? Then what are the differences between necrosis and apoptosis? Let us try to understand that briefly so that we never use these two terms interchangeably. Apoptosis is defined as a programmed cell death. That is, there is elimination of unwanted cell. Now remember that the body can eliminate unwanted cell in its normal physiological state or in a pathological state. But necrosis is almost always pathological. In case of apoptosis, the cell always shrinks, whereas in necrosis, the cell usually enlarges. In apoptosis, the nucleus fragments itself into very tiny nucleosomes. Whereas in necrosis, do you all remember what happens to the nucleus? The nucleus in necrosis undergoes one of the three changes. One is pycnosis, the other is karyorrhexis, and the third is karyolysis. In case of apoptosis, the remnants form apoptotic bodies and they are directly phagocytosed. So since they are phagocytosed, there is no inflammation involved. But in necrosis, the plasma membrane disrupts, there is leakage of content, and that usually triggers inflammation. So I hope you all enjoyed this video on necrosis, and this video gave you all an insight or two into the topic necrosis. And if you all did, and if you all would like to see more such videos, please like, share and subscribe to my YouTube channel. And here's wishing you all a very happy learning. See you all till the next video.